পুরো কতগুলো
17 শুরু করি।
তোমার ফোন কোথায়?
ফোন চার্জ হচ্ছে। ফোনে কিছু করিস না এখন? যাচ্ছি না।
দেখো আমার সাথে বেরোবো।
Okay. Good evening to all of you. On behalf of the Department of Conservation Biology, I welcome Dr. Subarna Thakur of Department of Bioinformatics, University of North Bengal. She has a subject specialization on comparative genomics of bacteria, but apart from this, she has several research experiences in microbial comparative genome analysis, metagenomics, microbial genome evolution, and many such other things. Before joining NBU, she was associated with the University of Angers, France, under the Rose Genome Project, and she was a research associate in Bose Institute, Kolkata. So I hope we will have a very good learning experience today. So over to Madam. It's your class now.
Yes, thank you, sir, for the introduction. So before moving on, as sir told you that my name is Dr. Subarna Thakur, and I'm currently working as assistant professor, and I'm also the head of Department of Bioinformatics in University of North Bengal. So I have been asked to convey this lecture on the basic topics of bioinformatics, since you have few things in the your PhD coursework, and many all of you are doing your PhDs in various fields of conservation biology. So you should know the basics of some of the tools that are commonly used for the bioinformatics. So today, as per your syllabus, so I will discuss about the various databases that we have, along with that how to retrieve information from the database, that include database searching, and we will also talk about the common data formats that you will come across in the database. Along with that, how you use this data for sequence alignment, and after that maybe we'll proceed to how to construct the phylogenetic trees and the structures prediction and all those things. So let me first go to the basic topic of the biological databases. So as many of you must know that bioinformatics is a very interdisciplinary field, and it comprises of computational elements along with the biological data. So basically it is a field which deals with the analysis of the biological data. So when I talk about biological data, the first thing that come across is how do you get these data? So maybe there are different kinds of experiments that are going on over the years in several kinds of laboratories. So they generate a lot of data for us. Data in the form of genomic data, or it can be proteomic data, it can be gene expression data, it can be even some published literature data, it can be medical informatics data, it can be immunoinformatics data. So there are different kinds of data that are generated from the laboratories. Now you need to store, analyze these data in a very meaningful way. So there comes the importance of the computers, particularly the field of the computational biology and bioinformatics, which deals with this kind of storage of the data. So basic premises that we are talking about is the data and how they are stored, and what are the tools that are needed to retrieve those data, and of course the tools that are needed to analyze those data. So this is basically the premise we are talking about. So moving on to the topic of the databases. So before moving to the actual topic of database, let me first share the screen with you, and then maybe I can proceed with my talk. So first of all of you can hear me, I hope.
Yes, ma'am.
Is my audio clear to all of you?
Yes, ma'am.
Okay, so thank you. So as I move along, so let me first share my screen with you. That will be good. Just give me 1 second. Yes, so the slides are visually available for all of you.
Yes, ma'am.
Okay, so we'll move across. So what I was talking about is the database. So databases are basically a kind of a computational archive, or you can call it a digital library, which is used to store and organize data in a very suitable way so that information can be retrieved whenever you want to. So database is a collection of this interrelated data which are stored together. And another thing is that the data should be stored in such a way that they can be retrieved later. So this is the first thing, is that you should store the data in such a way that you can retrieve those data whenever it is required. So if I tell it in a very simple way, so I would call it a kind of a digital library. Okay, see, all of you are aware that library consists of books, and the books are arranged in a proper way so that you can always get information out of books. So similarly, here we are talking about these data which is equivalent to the books, and these data are stored in a very proper way, in a very organized way, in a digital library, and that is what the database is all about. So if I call it a database, if you see in computational terms, it is nothing but a series of tables. So it will be something like a table, but it is interconnected tables where you will have the fields, you will have the actual records in terms of row. So all of you are aware with the Excel sheets, right? Microsoft Excel sheet. So if you open a Microsoft Excel sheet, you will find something like in rows and columns or something like this. So it is a kind of you can say a kind of a file with number of multiple entries where you can you can put it across the data in these tables in multiple rows and columns. So just imagine that this is something similar to that, but this is not like Excel sheet, but this is created with the help of certain tools, certain other tools. But this is a kind of a tables which are stored digitally, and this table comprises of a number of columns, and the number of columns may have various rows which will hold the actual entries. So this is the basic premises of the organization of the database. So I will just skip this part, but basically what is that there are certain management softwares which are known as the database management softwares, but these are softwares which help to manage your database so that you can store your data in a proper way. And another thing is that whatever you store the data, there is multiple tables in a database. So all the tables are stored in a way so that they are interlinked. So whenever you have a query, so you will get the information in terms of interconnected data. So these tables that are stored are all interlinked with each other, and this kind of a management system is required for this kind of thing. So there is a database management system that does it for you. So moving on to the actual role of the databases in biology, why do you require those databases in biology? So I have put across some of the important points that you should always remember, that what is the need of having the databases in biology. So the first thing is that there is a need to store and communicate large data set. As I just told you that in the various parts of the world, there are a lot of big projects that are going, projects in sense of genomic project, proteomic project. All of you are aware of the Human Genome Project, right? So there is a Human Genome Project that was completed in almost in 2000 and 2001. So it was a big project, and from the Human Genome Project, of course, then the era of genomics came, and there was a lot of sequencing of the genomes that are going on. You will find across almost all the higher plants and animals, their genomes have been sequenced. Even for bacteria, genome sequencing has becoming very easy. Every day we are coming across new genomes of bacteria. So there are this huge experiment that deals with this genomic data, and there are a lot of data that are coming from this genomic project. Similarly, we have data coming from the proteomics project and from metagenomics project. There is a new field that is coming up, which is metagenomics, where you directly get the genetic material out of the environmental samples without going for culturing of the bacteria in laboratory setups. So this is a metagenomic concept that is coming up, and this huge metagenomic projects are being going on, where they are going to extreme environments like they are going underwater, or they are going to this thermic vents, they are going to volcanic rocks, and they are getting the samples out of this extreme environment, and then they are directly going for the genomic sequencing without culturing those bacteria in specific laboratory. So this is a metagenomics that is coming up. In fact, there is a huge project that is going on over the years, which is known as the gut metagenomics. Sorry, I have a very bad cough. But as I was talking about, there is a huge project that is coming up, and it is called the gut microgenomics. And what they are doing is that they are trying to get the DNA sequences out of the gut microflora. So we have different kind of bacteria that are growing in our human gut or digestive system, and if you try to culture those bacteria in environmental conditions in laboratories, it's never possible. So what they are trying to do is that they are trying to get the genomic samples out of the digestive system, and they are trying to get the genomic information right out of the sample. So that is a gut microflora metagenomic project that is going on. So you can imagine that these projects will generate a large amount of data which are impossible for us to analyze and store them manually. It's never possible. So of course you need big computational powers to analyze those data, to store those data. So there is a large data set that are being generated over the years, and it is growing day by day. So of course you need the databases to store this data. And of course there is a need to disseminate the biological information. It's not just storing, it's a thing that the data are coming in public domain. So it is easier for us to get access to this data. So suppose I have a big experiment that is going on in some part of the US. So I may not be aware with the experiment. I'm not, of course, I don't have information across over the whole world that what kind of experiments are going on in each lab. It is never possible. But it is always a mandatory thing to now to publish any experiment or publish any data. You have to put the data in databases so that they are available in the public domain. And whenever they are available in the public domain, everyone has access to this data. So this is a growing trend that whenever you are doing certain experiments, you need to give the results, you need to give the data in public domain, so everyone has the access to your data, so that they know that what kind of experiments have been conducted, what kind of experimental results are being generated, so that you can of course get information out of that. So this is a kind where, and that is where the importance of databases in biology comes. And of course, the data that need to be stored in these databases have to be in a format which can be read by the computer. So you can't just store any data in any format, okay? There need to be some kind of special format which can be read by the computer, because of course these databases are all handled by computer softwares. You need to make those databases with the help of certain software. So there is this computers which need to read those data. So you have to prepare your data in a readable format. This is very important, that you prepare your data before publishing your data in the database or giving it to the database. You need to make your data in a proper format, in a machine readable format. So the computer knows that this is the data, and it can store and it can analyze those data for you. So this is very important. So as we move along, let's look at into the different classifications of the databases. So according to what kind of data that are being stored in the databases, so we have different kind of databases. So some of the databases are the nucleotide sequence databases, we have protein sequence databases, then there are databases which deal with the three-dimensional structures of protein. Now all of you must know that proteins are of course in three layers of organization, right? The three tiers of organization. First we have the primary sequences, that is comprising of the amino acids only. Then we have the secondary, where we have the secondary structure information in terms of loops, motifs, domains, and other kind of thing. And of course the third layer is the third tier is the three-dimensional structures of proteins, which of course ensure that how the proteins will function. So there are these three layers of protein organization, and there are certain databases which deals with the three-dimensional structures of protein. These are known as the structured databases. So these structured databases are where you can get the three-dimensional structures of a protein, and you can use them for your protein structure prediction, or you can visualize the proteins. And of course then we can go for some kind of other important aspects like computer-aided drug designing and docking and all those things. So this databases, they houses the three-dimensional structures of protein. Other than that, there are databases which deals with the gene expression. Gene expression means that how a gene is expressing under certain condition. So previously there used to be this microarrays. So this microarray experiments would result in lot of data, and that microarray experimental data were stored in these databases. Now of course we have the RNA-seq data or RNA sequencing technology data, which tells us a lot about the gene expressions that are going on in a particular condition or experimental condition. So this there are certain databases which deals with this kind of expression data. Then there are databases related to the metabolic pathways like the KEGG. I will talk about the KEGG database, but for the time being, let's see that there are certain databases which deals with metabolic information system biology. So there are databases specifically dedicated to that. And of course, there is databases which houses information related to proteomics, like the MSMS data or GCMS data or some kind of other proteomic experimental data, maybe mass spectroscopy also. So there are certain databases which which has this basic proteomics data in them. So what kind of data ensures that you have what kind of database? So according to the data they store, we can have a lot of different types of the databases. There is another way by which you can classify the databases. One is the primary database, then we have the secondary database, and another is the aggregate database. So the primary database include the data which are directly coming from the experimental setup. So if you do any experiment and you generate any data, and you can directly deposit in your data in these databases. So all the nucleotide or protein sequence databases are generally the primary databases. So if these databases are only housing data that are coming directly from the experiments. Now we have another type of database which are known as the secondary database. Secondary database will have data which are generated as a result of the analysis done on the primary data. For example, if we have a amino acid data. Okay, suppose you are doing certain experiment and you have generated certain data that are related to amino acids. What you can do is that you can do certain analysis where you can run certain tools to analyze the secondary structures of the amino acid sequences. So you will try to predict the loops, domains, or conservative motifs out of this amino acid sequences. And if you get any result, you can put them into another database. So that will be a secondary database. So you can see that the secondary database will houses information that is as a result of the analysis that was done in the primary data. Okay, so that is the basic difference between the secondary and the primary database. So there are some examples of secondary database which I have put together. We have the TREMBL, we have the PFAM, we have the ProSite, we have the Profile, SCOP, CAT. These are all different types of secondary database. Particularly this PFAM, SCOP and CAT deals with the different secondary structures and their families of the proteins. So these are basically the databases which have information about the protein families and the secondary structures of the proteins. So these kind of databases are known as the secondary database. And the last type of database according to this kind of classification is the aggregate type of database, which will have a mixed of the information. So it will have a combination, or you can say a consolidated part of the data where it can have both the primary data and the secondary data. So it is a mixed bag. Okay. So there are certain big databases like the database that we see in NCBI or EMBL. So these are big databases which can both be a primary database or it can be a secondary database. So it is a kind of a mixed kind of a thing. So this is the databases that we can have. So this is basically the different ways of classification of databases. So another way, of course, is to based on the availability that if your database is publicly available or not, or if they are academically available, based on these categories also, sometimes we classify the databases. So one thing I must tell you that there are these big databases like the NCBI. So let me ask, has anyone used NCBI before? I'm just asking all of you, do you have any idea of the NCBI database? National Center for Biotechnology Information Database. Have you used any of you? Sorry, can you hear me?
Yes, ma'am.
Yes. So have you used the NCBI database? I'm just going on the NCBI. I'm just coming back to the NCBI database. So have you, any of you used the NCBI database?
I haven't used it, ma'am. I don't know about the rest of the people, but I haven't.
Okay, so I'll just give you a glimpse of the NCBI because it is one of the most commonly used database that we have. So this is the, can you see the window? Is it visible to all of you?
Yes, ma'am. Yes, ma'am.
Yes, yes. So this is the NCBI database. So uh, many of you, of course, will proceed to do your PhD, and if you work in any kind of, even in wet lab, not only bioinformatics, but even if you work in any kind of a molecular biology or cell genetics or something like that in any field, you will of course certainly have to use this database. This is one of the biggest database that we have. It is the NCBI database. NCBI is the National Center for Biotechnology Information, and it is being maintained by the US government, particularly the National Institute of Health in US. They maintain this database, and this is a big database which has a lot of information related to sequences, nucleotide sequences, protein sequences, expression data, genome sequences, and lot of things, SNPs data. So this is a very big database, and you will, all of you, I'm certain that if you go for your PhDs, at one point of your time, you will have to use this database. This is a very important database. Please remember that, NCBI database. And it's not only that, it will have the information. It has very important tools also related to how you use the database. So it has tools like the BLAST, which I'll proceed to. So this BLAST tools is also connected with this database, and this database is a very important database, and it's a big database which has a lot of sub databases also. So this NCBI is important in this aspect. So that is why I just wanted to tell you about this database. So moving on, let me go back to the nucleotide sequence database. So there are certain databases which deals with the nucleotide sequences. So the one that I showed you, NCBI, this NCBI is housing the current nucleotide sequence for the US government, that is the gene bank. So gene bank is a database that is for the nucleotide sequences, and this gene bank is now linked with the NCBI database only. Then there are certain other databases like the EMBL. Then we have the DDBJ. So EMBL is a database which is maintained by the European Union. So they have their own sequence database, and EMBL stands for the European Molecular Biology Laboratory, and it is a database which is maintained by the European government, European Union basically, because the NCBI, which I showed you, is of course being maintained by the giant US. So US are maintaining the NCBI database, and the equivalent of that is the EMBL database, which is maintained by the European Union. From the Asian part, we have the DDBJ, DNA Data Bank of Japan. So as the name suggests, it is coming from the Japan, and Japan is maintaining this sequence database. Now, you should remember one thing, that what I told you, if you are part of any genome sequence project or genome project, or if you want to publish a new gene, maybe you are working with a new gene or something. So if you want to publish that validly in any scientific journal, it is now mandatory that you have to submit your sequence in any of these sequence databases. So if you submit your sequence in this sequence databases, they will of course provide you an accession number, and this accession number is generally mentioned in your published literature. So this is becoming mandatory, that if you are working on any new gene or even a genome or even a partial gene, you have to submit your sequences in any of this database. Okay, you can submit it in NCBI or you can submit it the EMBL, that doesn't matter, but of course you need to submit your sequences in this sequence databases. So these are a kind of a primary database that is there, that is the EMBL, Gene Bank or DDBJ, that is, I already told you about this primary databases. So I'll move to certain other databases, particularly with the databases related to genomics, that is, it houses information related to genes location, and there are certain databases which are dedicated to a particular organism. Okay, for example, it can be human, it can be Arabidopsis, or it can be mouse, it can be yeast. So there are certain databases which are dedicated to a particular organism, and it will house all the gene related information of that organism. Okay, there are databases which are particularly dedicated to the plant Arabidopsis. Okay, Drosophila is also a very well known fly, and it is known as the fruit fly. All of you must know about the Drosophila, and you know that there are a lot of experiments over the years which have been done on the Drosophila. There is experiments related to genetics, mutations which have been done in Drosophila. So there are certain databases which only houses information related to Drosophila, any kind of information, any kind of genomic information in terms of genes location. Then there is SNPs or single nucleotide polymorphism. So there are a lot of databases like that. So these are databases which are dedicated to the genomics of maybe a particular organisms only. So I'll move to another kind of database, that is the protein database. So previously I talked about the nucleotide databases. So you will have the nucleotide in terms of gene sequences. There can be other type of database, which is the protein sequence database. So if you are dealing with the amino acid sequences, so there are databases like that. There is SwissProt, then there is Interpro, there is like IntIns. So there are a lot of different. IntIns is a database which houses information related to the enzyme. So if you have any information related to enzyme kind of protein, so that will be housed in IntIns. So if you are looking for any particular kind of information, you have to look into these databases. So if you are interested in nucleotide sequence databases, so you will go for the NCBI or Gene Bank or something. If you are interested in protein sequence databases, you have to look into SwissProt, Interpro, or something like that. So these are the different databases related to sequences, protein sequences. Then of course I just talked about the structure database. So as I was telling you about the structures, because we know that proteins have these three-dimensional structures, and there is a database which is known as the PDB, Protein Data Bank. It's Protein Data Bank database, so PDB database. So it is a database which houses information related to the three-dimensional structures of protein. Generally, three-dimensional structures are resolved by certain experimental procedures like the excellar crystallography, NMR, or maybe cryo-electron microscopy. Okay, so there are certain experiments which are going on for determining the three-dimensional structures of proteins, and whatever information that are retrieved from these experiments will be stored. So it will be stored in this particular kind of databases, which is the three-dimensional structure databases. And there are certain other databases which deals with the structural features of proteins. So there is DALI, there is FSPP. So there are a lot of different kind of databases which are dealing with the structural part of proteins. Okay, since proteins are three-dimensional structures, so of course there are databases which deals with that. Other than that, there are few databases which deals with the structures of RNA. Okay, now all of us know that RNAs might have a typical structure like a cover leaf structure, or they might have certain kind of other structures. tRNA has certain kind of structure, then we have the ribosomal RNA. So there are certain databases which deals with these RNA structures also. So whatever we are talking about in terms of the 3 dimensions. So if you are dealing with that kind of data, so 3-dimensional structure, so then we have to look into this kind of structured databases. Okay, so now let's move to a very important point, that what kind of file formats that you will come across in these databases. Okay, now whenever you retrieve any information, or even if you want to store any information also, that has to be done in a particular format. Okay, because you just can't use any format. Okay, if I suppose I want to submit my data in one database, so I can submit my data in one format. Suppose another person comes, he will submit his data in another format. So there can be a lot of confusion regarding that. So there has to be a proper way by which you can submit your data in a database. Similar way, if you want to retrieve your data from the database also, that has to be in a particular format. Okay, so there are certain formats which are very important in bioinformatics. Another thing is that because whenever you retrieve your data from the database, make sure that you may need those data for further analysis. So you maybe have to run certain other softwares on those data. So your data has to be in a proper format. Okay, you can't just have data in random formats. There has to be certain formats which can be recognized by the softwares. So there are certain popular formats which are very well known, and whenever you retrieve or you submit your data in these databases, that has to be followed. So there is this format which is known as the ASN format, then there is EMBL format, then there is FASTA. I will talk about the FASTA format because all of you should know about this format, because this is the most commonly used file format. Okay, FASTA, F-A-S-T-A. So FASTA is the most commonly used file format. Most of the databases have this format. So if you just go to any database and if you want to download any relative information, so most of the databases, particularly if you are interested in genomic data or protein sequence data, most of the data will be retrieved in this particular format, which is known as the FASTA format. Other than that, we have the GCG format, gene bank format, FileLib format. So there are a lot of formats that are being followed, but the FASTA is perhaps the most important format that we'll have. So before moving to the FASTA, I just wanted to give you a glimpse of some of the important database. So NCBI, I just showed you the NCBI. This is the home page of the NCBI. So if you can type NCBI in the Google, and it will show you the page for NCBI. So NCBI is one of the most important or most biggest biological database that we have. And of course, I told you that is maintained by the US government. Other than the NCBI, there are certain database I just told you about the gene expression. So there is this database which is known as the Gene Expression Omnibus. It's called the GEO, Gene Expression Omnibus. If you just write in the Google GEO database, it will of course then give you the link for that. So this is a gene expression database. So it will have information related to the gene experiments, gene expression experiments. So it will have experiments like microarrays, or even now RNA-seq data are also there in this kind of database. So these are some of the important. I'll just skip those parts. Let me go to another very important aspect, that is the file formats. Okay, so I just told you, so I discussed about the file format. So let me go back to the file formats that I was talking about. Okay, so the file format. Yes. Just give me 1 minute so that, yeah. Okay. So, as I was talking about, there are several formats by which you can retrieve any sequence. So suppose I want to retrieve the nucleotide sequence of any particular gene. Okay, let me give you an example. Suppose I want to retrieve the gene information of the NIF gene. What is a NIF gene? NIF gene is the nitrogen fixing gene. Okay, so there are certain bacteria like the rhizobium, and they have this special kind of gene which will code for the enzyme nitrogenase, and this enzyme will ensure that it can fix the atmospheric nitrogen. So NIF gene is something which is found in few of the bacteria. So I want to retrieve the nucleotide sequence of the NIF gene. So I'll go to the NCBI and I will try to find, there is a search option. I can just type in the search option NIF gene and it will retrieve my data. Now the question comes, how does I retrieve my data? There is a file format which is generally followed, and this file format is commonly known as the FASTA format. So try to look into the aspects of the FASTA. So typically your FASTA will have this kind of thing. Sorry, I'll just go back. All of you can see the pointer, the FASTA. So this is the typical FASTA sequence. If you look into this slide, so this is a typical FASTA format that you will come across. And any FASTA file will have this symbol, which is a greater than symbol. So most of the FASTA information starts with this symbol, which is the greater than symbol. Then comes a number. Generally this number is the accession number. Okay, so accession number is something, a kind of a number which is generally being given to each of the sequences that are deposited in this database. So this is which is known as the accession number. It is something which is similar to the accession number that you will have in normal libraries. Okay, so every books in libraries have certain kind of accession number, and if you go to a library, you can search a book with the help of any author or the title of the book, and if you don't know any of the information, you can just search your book with the help of the accession number, right? So it's just imagine similar to that. So if you don't know about the gene and what kind of gene it is, but you know that this gene might have this accession number, so you can search any database with the then symbol. There will be this accession number for this particular gene, and then there will be information related to gene. What kind of information? What kind of organism from which it is derived? What is the product of that gene? Okay, so that information will be in the first line. So the first line is actually the descriptive line of the sequence. So it starts with a greater than symbol, and then we will have the single line of description about this gene. And that will be followed by the actual sequence. So if it's a gene sequence, it will have this A, T, G, C, 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 like that. Now all of you know that nucleotide data will only comprise of this 4 letter A, T, G, C. So your nucleotide sequences only will have this 4 letters or 4 alphabets, that is A, T, G, C. And if it's a gene sequence or nucleotide sequence, all your information will be in such A, T, G, C, 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 like that. Okay, and if it's a protein sequence, then of course we'll have the 20 different letters for the amino acids, and these 20 letters will be joined together, and they will have this information in terms of the amino acid sequences. So this is the information that is there, that will be there in the FASTA format. So FASTA is the most commonly used format for the database, and most of the database will have this kind of FASTA file. Okay, so if you retrieve your sequences from the database also, it will be mostly in this FASTA format. So this is a very important format of the database. So I just wanted to tell you about the FASTA format. Other than the FASTA format, single line FASTA format, we can have the multi-FASTA format. What is that? Multi-FASTA format means that within a single file we can have multiple sequences. Okay, multiple sequences. For example, look at into this. So this is a multi-FASTA format, means that there will be multiple FASTA sequences in a single file. Okay, so this is a random which I put together. Don't interpret it like that, Jhuma, Bhuma. It's nothing like just, it is just randomly I have put. So what this is that you can have multiple FASTAs, means that you can have multiple sequences in a single FASTA. Generally, this kind of formats are used for sequence alignment. Just a moment, I think I have a call that is coming on. Just give me 1 minute. Okay, just give me 1 minute. Okay, I'm just muting you. Sorry for the interruption. I had an important call, so sorry. So as I was talking about, so let me again share the screen with you. All of you can hear me?
Yes, ma'am.
Okay, sorry for the interruption. So I'll just share my screen with you again. So this is the multi-FASTA format which I was talking about. So generally this kind of formats are used for phylogenetic tree construction. So when you want to create evolutionary trees, or if you want to create phylogenetic trees, you need to compare your sequences and you need to do sequence alignment, which I will talk about sequence alignment also. That part also I will discuss. But you need to do sequence alignment, multiple sequence alignment. In that case, you need to have this multi-FASTA format of the files. So generally this kind of formats are used for doing a lot of downstream analysis of the sequences. So this is one important format of the data by which you can retrieve your information from the database. Other than that, I told you about other type of sequence database. Sequence data is the gene bank data format. Okay, so this is one format of which is generally the sequences retrieved from the NCBI. So if you go to the NCBI, so you can retrieve your information in this gene bank format. So FASTA is always there, but other than FASTA, you can also retrieve retrieve your information in the gene bank format. So generally the NCBI has this format, NCBI database, which I showed you earlier, the National Center for Biotechnology Information. They will have this format. So what is a gene bank format? You can see that this is a sequence. This is a particular sequence which I put in the screen. This is the insulin gene, insulin mRNA gene from the human. And if you search this sequence and retrieve your sequence in the gene bank format, so generally your gene bank format is a very large format, and the first part of the gene bank format, we will have information related to the sequence. What kind of information? Like what is the organism name, who are the people involved in sequencing that particular gene, and if that gene has been published in any kind of journal, and if it is having any kind of accession number. So generally the accession number is in the topmost part. Accession number, which I told you, is unique to each of the sequences. So if you submit your sequence any database, so that will be provided in, they will provide a gene bank number or an accession number to your sequence, and generally the accession number is in the topmost part, and rest of the will have a descriptive information about your sequence. So they will have a more descriptive kind of information in this part. And if you go to the down part, this is a particular part. I have just put, all these are in a single file. I have just put it in 2 slides for you to understand. So the first part we will have the descriptive part, and the next part will actually have the information related to the sequence. So you will have the sequence in the form of, if it is a gene, so the number of nucleotide, like the first 285 position is the actual CDS or the coding sequence part. So you will have that information. Then you will have the information like what is the role of that gene, that is also there. Then if it is having any kind of signal peptide or something like that, that will be also there. So a lot of features related to the sequence, that information will be present in this middle part. Okay, so the first part is the descriptive part, next part will be the feature part about the sequence, and the last part we will have the actual sequence. So the actual sequence, actual gene sequence of that insulin which I showed you is this one. So, and you can see at the end there are 2 backslashes. So this marks the end of the file. So what you saw in FASTA is that FASTA has this unique feature of greater than sign. So if you look at the FASTA, it is marked by the greater than sign, and if you look into this file, these have a typical backslash like thing at the end. Okay, so this marks the end file, end of the file. So this is a kind of a file format which is commonly known as the gene bank file format. This is generally found in the NCBI. So if you go to the NCBI and if you search any gene sequence, so you have these 2 options. You can download your file in either gene bank format or in FASTA format. So you can retrieve your information in these 2 formats, which is the gene bank format and the FASTA format. These 2 format is further recognized by a lot of softwares for doing a lot of different kind of analysis, like the phylogenetic tree construction, or maybe some kind of other if you want to do the annotation, or maybe you want to do for the any kind of mutational study. So that kind of software, they will recognize their input in the form of these 2 formats, gene bank format or FASTA format. Most of the downstream analysis softwares, they recognize these 2 formats, that is the gene bank format and the FASTA format. Remember, always remember, FASTA is a very common file format that will be seen in many of the database. Acha, now I will move to a very important topic, and I will restrict my lecture with this topic only, that is the gene bank searching. That how do you search the gene bank? How do you retrieve information from the gene bank? For that, we have to use a particular tool that is known as the BLAST. Okay, so I will mention about the BLAST, and I will also tell you where to look for the BLAST, like where you can find this tool. Okay, so let me first give you an introduction of the BLAST tool. So for that, I will share another screen. Okay, just give me 1 minute. Yeah, BLAST. So what is a BLAST? BLAST is the basic local alignment search tool. Okay, basic local alignment search tool. It is a search tool that is commonly used for searching database. So suppose you want to search for any particular sequence. So what, or if you want to search, if your particular sequence is similar to other sequences that are already there in the database, then you have to perform this BLAST analysis. So for example, suppose I retrieve a NIF sequence, okay, nitrogen fixing gene sequence, which I told you, and I want to find certain other sequences which are similar to your NIF gene, maybe from other bacteria or maybe from other kind of nitrogen fixing bacteria. So I want to retrieve similar sequences. So if you have one sequence and you want to retrieve similar sequences from that database, then you have to use this BLAST tool. So BLAST is a kind of a tool, a similarity search tool that is commonly used for the databases. Okay, so BLAST is a tool. It is particularly related to the NCBI database. So if you have if you go to the NCBI and you want to find similar sequences in the NCBI, you have to use the BLAST. So I'll quickly move to the actual part, like what are the different what is the logic behind the BLAST. So what you do is that, so they have a form, they have a web page where you can submit your query. Query here will be means that you have to submit any sequence for which you want to find similar sequence. So suppose I tell you that I want to find a NIF sequence. Okay, I want to find a sequence which is similar to the NIF gene. So I will paste my sequence in this query page. And remember, you have to paste your sequence in FASTA format, which I earlier told you that generally the FASTA is the most commonly used format. So I will ensure that my sequence have to be pasted in the FASTA format. So then there is this query option. You can just submit your query, and this, once you submit your query to that, there is a BLAST web page, and the BLAST web page will connect to a BLAST server. So BLAST server is the one computer which is having this algorithm to run the BLAST. It is located remotely in some part of US. So your BLAST page will connect to the BLAST server and will do the analysis. That, like, analysis here means it will try to find similar sequences to the one that you are searching. So they will retrieve the sequences, and once it is retrieved, then the returned results will be again sent to the web page and it will be displayed over here. So that is the basic logic by which you can use this basic web tool, that is the BLAST. Now remember, this is the BLAST is in two formats only. It can be a web page format also, or there is a standalone version also where you can download the BLAST and install the BLAST in your computer, in your local computer also. But most of us use the web page, so that is the basic thing. Now what I will do is that I will directly go to the web page to make you understand how the BLAST works. So let me go back to the web page of the NCBI. So if you type NCBI in Google, so it will of course redirect you to this web page, that is the NCBI web page. Okay. Now if this is the home page of the NCBI, and in the lower end you can see that, please look at the cursor. Okay, there is this BLAST. Okay, so if I click onto this BLAST, just click onto this BLAST, and it will direct me to this home page, that is the BLAST. Okay, so this is the home page of the BLAST tool, and as you can see, this basic local alignment search tool, this is a tool that is used to find similar sequences in the database, in the sequence database, particularly sequence database. So there are few options of BLAST for me. One is the nucleotide BLAST, then we have the protein BLAST, then we have the BLAST X, and another option is the T-BLAST10. I will explain each of you. Now the first one is the nucleotide BLAST. What is a nucleotide BLAST? So suppose you have a nucleotide sequence, and remember your sequence should mostly be in FASTA format. So suppose you have a gene sequence that is in nucleotide sequence and it is in FASTA format. So you want to retrieve certain other gene sequences which are similar to your sequence, then you have to go for the nucleotide BLAST. So here your query is also nucleotide, and the results that will be retrieved from the database will be also a nucleotide sequence. So that kind of BLAST, when nucleotide query and nucleotide database are used, that is a nucleotide database. Other type of BLAST is the protein BLAST, where your query will be protein sequence in terms of amino acids. So your query will be amino acid sequences, but remember your amino acid sequences should also be in FASTA format. So you have to paste your amino acid sequence over here, and it will try to retrieve similar protein sequences from the database. So here your query is also protein, and the sequences that are retrieved from the databases will be also protein. So this is protein BLAST, or commonly known as the protein BLAST. Now there are two other BLAST which are sometimes used, not commonly but sometimes used, that is the BLAST X and T-BLAST10. So BLAST X, what is that? Here your query sequence will be a nucleotide sequence, but it will be translated to a protein sequence and then searched against the protein database. So here your query will be a nucleotide and your database will be a protein type of database, sequence database. Okay, so here what my objective is to find if I get a nucleotide sequence, I will try to find sequences which will code for same kind of protein as my nucleotide sequence. That is my basic logic behind doing this. This is mostly done in case of annotation, okay? Gene annotation, genomic annotation, where you try to find the possible products of your genes. So what kind of protein products will be produced from a particular gene? If you have to guess that, so you have to go for the BLAST X. So here your query is nucleotide, but your database that is searched against will be a protein sequence database. So query nucleotide and protein sequence is the database. So that is the BLAST X. Just opposite to that is the T-blastane, where your query sequence will be a protein sequence in terms of amino acid, which will be searched against a nucleotide sequence database, but each of the sequences in the nucleotide sequence database will be translated into a protein sequence for the search. So here your query is protein and it will be searched against a translated nucleotide sequence database. So these two are a bit critical kind of BLAST, but BLAST X is mostly used for annotation purposes, but we mostly use the nucleotide BLAST and the protein BLAST to find similar sequences. Okay. Now let me go back to the BLAST page, actual BLAST page. So let's see the nucleotide BLAST. If I click the nucleotide BLAST, it will give me this kind of web page. Okay, this is a web page that is known as the nucleotide BLAST kind of web page. So here you can see is that the first thing is that there is a query box, or this is a part where you can paste your query sequence, and that has to be mostly in FASTA. You can see it is written, it is mostly your query sequence has to be in FASTA format. So if you have a gene and you want to search similar sequences from the gene, so you need to get the actual sequence of that gene in FASTA format. Okay, so for searching you need to get your sequence in FASTA format. So here you can paste your sequences in FASTA format. And also other thing, if you don't have the FASTA format, what you can do is that you can also give the accession number. What I told you about the accession number is that every gene that is submitted in the database, we will have that in the form of they will give an accession number. So accession number is unique for each gene. So if you don't get the actual sequence also, if you know the accession number, you can use the accession number also. Where you will find the accession number? You can find the accession number from the database, or sometimes in some published literature also. If you find any scientific paper in the journal, so sometimes they mention the gene sequence in terms of the accession number. So accession number will be mentioned in that sequence. So you can retrieve the accession number from published literature also. So you can paste that accession number and it will do the same thing as what it done for the FASTA sequence. So once it is done, if you come to this part, this is actually the database searching part. So you have to search it against a database. So there is an option over here, like what kind of database you want to search against. Okay. So the most by default option is the nucleotide database, or the NRNT database. So this is the standard one, but there are certain other options also which you can use if you want to use it against the RefSeq sequence and RefCheck genomes and lot of other things. So there are certain other options for you, but by default, if we just want to search similar sequences, so that is the nucleotide database that is commonly used, that is the NRNT database. Okay, NRNT database collection. So this is one thing that is commonly used. And then there are certain few other options. For example, if you want to restrict your search to any particular organism, for example, I want to restrict my search to humans only. So I will write the Homo sapien. If you want to restrict my search to any mouse sequence, so I will write the Rattus or the scientific name of the mouse over here, and it will restrict my search result to a particular organism. Okay, so this is something which can be done. There are a few other options which I'm not going around. And the last thing is that you have to click into the BLAST. If you click into the BLAST, this one, after a few seconds, end, it will give you a result page where you can see the actual similar sequence to the sequences that you have used as query. So suppose I have a sequence and I want to search similar sequences from human beings or from the mouse. So then a list of these sequences will be produced in the result page. Okay, so this is how you can use the BLAST to look for similar sequences. So other than nucleotide, you can use the protein also, protein search option. Here only you can go to the BLAST page, BLAST P page, and it is something similar to that of the BLAST N. Here, but one thing, for the nucleotide BLAST, you need to paste the nucleotide sequence, but here you need to get the amino acid sequences since it is dealing with the amino acid proteins or protein sequences. So you need to get your FASTAs in amino acid format. So this is something which can be done. Okay, and you can paste your sequence and you can search your sequence against a particular database. So here the default option is NR, or the non-reductant protein sequence, that is NR, but you can have certain other options also. You can choose certain other options for searching the proteins, similar protein sequences. So there are a lot of other options as well. So generally by default, we generally use the NR database. Then of course, there is algorithms like what kind of BLAST you want to use. The most common one is the BLAST P. Other than there are certain sophisticated BLAST like PSI, PHI. These are more kind which is dealing with a lot of secondary structure information and all. So I'm not going into that, but basically generally we want the BLAST P. We use the BLAST P option here, and then we can use the BLAST button over here, and as you click onto the BLAST button, a result page will show you sequences similar to your amino acid sequence. So this is basically a page or a tool, a web page, which is used for finding any similar sequences to a particular sequences. Okay, so if you want to find similar sequences to a particular gene or a particular protein, so you have to use this. This is a database searching tool that is commonly used, known as the BLAST. Okay, so that is what the tool that I wanted to tell you. I wanted to show you some other things that which I perhaps skipped, like how to retrieve the sequence. Okay, now let me go back to the NCBI part again. So let me show you how you can retrieve your sequences. Suppose I told you that I need to get my sequence in the FASTA format. So how can I retrieve my sequence in a FASTA format? So let me see. Suppose I want to retrieve a gene sequence in maybe the NIF gene. Suppose I want to retrieve my NIF gene sequence in FASTA format. So what I can do is that there is a search option at the top, and in that top search button, I can write NIF, okay, NIF. And there is this options which are known as the database option, okay. So suppose what kind of database you want to search upon. Suppose I say that I will want to search NIF gene in the gene database. So they will search for sequences which have the NIF mentioned in the gene sequence. So let's see what I have just put NIF, and I have just put the gene option over here. Let me search it. Okay, so as you can see, what I have done is that I have just put that NIF and what it has done is that it has given me a lot of different options where they mention the NIF option over here. Okay, NIF option, anything related to NIF they have put. So it may be something in a NIF domain or something, wherever they have find the NIF domain mentioned or something like that, they have put together. Okay, so suppose let me now look into this. Okay, so what I will do is that I'll just click into this option. Okay, so if I click into this option, they have shown me that this is a particular type of a protein which have this particular domain which is known as the NIF domain, and it is from some fungus they have retrieved. They had this information that the NIF domain was present. This is not the nitrogen fixing gene that I was looking about, but they have found something which is similar name to that. That is why they have displayed. So I just wanted to show you how you can retrieve it. So I'll go forward with this particular gene only. So the page that will be shown to you is something like this. So this is a kind of a page which will have the information of a protein which is having this kind of a domain which is known as the NIF domain. So my motto here is to retrieve the sequence of this particular protein in a FASTA format. Okay. Now you can see is that there is an option called send to. Okay. Send to over here. And then there is this option called the file. As I go along, there is this option where I can give the information, I can retrieve this information in any kind of a gene table, or I can retrieve the information in any kind of formats, full report format or tabular format. So all this information which I see in this page will be downloaded to a particular table-like format or something like that. But if you want to retrieve the sequence in FASTA format, so I'll go down and I'll have these two options over here. You can see one is the FASTA, one is the gene bank. So if I want to retrieve the genomic information of this particular gene, so I can retrieve the information in FASTA format. So if I click onto that, so this is the information, this particular information, this particular gene information will be now retrieved in a FASTA format. So you can see that this particular sequence is FASTA, because how do I know it's FASTA? Because it starts with a greater than symbol, and then I will have the information related to this particular genomic sequence, like what kind of bacteria, what was the accession number, this NW0142 is the accession number, and then I will have the actual sequence over here. So the actual sequence is from here to here. So this is the information that is being retrieved in the FASTA format. Here also there is this option where you can download it in some coding sequence format, and if you want to download this particular thing in the FASTA, so there is this option where you can have it in a FASTA nucleotide or FASTA protein. So if I want to retrieve that in a FASTA file, so I will retrieve this in FASTA, and what I will do is that I will just create a file. So as soon as I create a file, a sequence will be downloaded in my local computer. Okay, so the computer that I'm using, a file will be downloaded over here, and I can just open that file in any Wattpad and Notepad. The same information that I showed you in the web page will be downloaded in my local machine as well. So this is the FASTA sequence, and this is how you can retrieve the FASTA sequences from the database. And of course, later you can use this FASTA sequence for searching similar sequences. You can use this FASTA file for further analysis, or you can use it for sequence alignment or anything like that. So this is how you can get the FASTA file from the NCBI database. So this NCBI database has lot of genomic information, nucleotide information, protein sequence information, and you can retrieve those sequences in either the gene bank format or the FASTA format. So I think with this I will stop the today's class. So I just give you a small recap of what I told you. So I told you about the various databases that we have, the protein sequence database, the nucleotide sequence database, then we have structured database, then we have databases related to gene expression, and we have the various file formats from which you can retrieve the information from the database. So I told you about the FASTA and the gene bank database, gene bank format, file format, and I also showed you BLAST tool. So if you want to search similar sequences from the database, you can search it with the help of the BLAST. It will be very commonly used if in this BLAST tool will be very commonly used for when you are working on molecular biology projects or cellular genetics projects. So suppose you have sequenced a new gene and you want to find similar genes from other bacteria. So then of course you will have to use the BLAST. So during the course of your PhD, you will I'm sure that all of you will use this NCBI database or the BLAST tool for doing works with your finding similar sequences or retrieving sequences. So this is a very integral part of the total PhD program that you will come across. So I think with that I will end my today's class. If anyone has any question, I'm very eager to attend it. So do you have any question? Please, you can ask the question to me.
Ma'am, I have one query. Can I ask?
Yes, sure.
Ma'am, we have learned to retrieve the sequences from these databases of nucleotide. Ma'am, suppose I'm not doing any work in molecular biology. For example, I am doing research in water quality and phytoplankton. Is there any way by which I can use this database?
Yes, of course. Suppose if you are working on some kind of conservation biology, as I told you, some kind of project. So there are these databases which have information related to a particular taxon. For example, if I go back to the NCBI, let me again show you this part. Maybe you can understand. Just give me the thing which I'll just show you. There is this option called a taxon page in the NCBI database. And other thing is that just have to go to the NCBI. Just give me 1 minute. I'll just go to the NCBI. And other thing is that the NCBI database can also be used for retrieving information related to some kind of published literature. Okay, it's not only gene sequence or protein sequences. NCBI can be used for searching. If suppose you want to find, if you have a plant, okay, and you want to see if that plant has some medicinal property, Okay, so what you can do is that you can search the literature which have mentioned that plant with some medical properties. Okay, so they will give you a lot of different experiments where they have used that plant in medicinal properties. So suppose you are coming across this new species of plant, okay, and you want to see if that plant is some medicinal properties or not. So you can use this database for that also. So searching for literature, published literature on maybe some ethnobotany related publishing or something like that. And there is this option called the taxonomy. So let me see. Yes, here. Yes, this is the taxonomy database. This is all there in the NCBI, okay? And in the taxonomy database where this can be used, suppose you have a plant and you want to know the position of that plant in plant system classification. Okay, you can just name the plant over here and it can search, it can give you the position, like under which order, under which family, under that, if it's a published one. Okay, something which is something totally out of blue, it cannot do. But if you want to, if you have any plant or you know the name of the plant, you can use this to get the taxonomic position of that plant. I think that is very helpful for the work that you are doing in your PhD. Of course, if you are coming across a new plant, you want to know the taxonomic position of the plant. Okay, so this taxonomy part, the taxonomy database will help you to nomenclature and classification of the organism. It can be used for plants, it can be used for animals also. So this NCBI is not only having information related to genetics or proteomics or like that, it is having a lot of information. I also told you about the information related to medicinal properties, and if it's published any information related to certain ethnobotany or something like that. So that will be also be there in this database. So you can always use the NCBI database for your research purpose.
Thank you, ma'am. Can you please show me again where can I find that tax?
Yes, taxonomy. Just give me one. I'll just give you one. Here, just go back to this part and you can see, this is the home page, okay? Just go back to this is the home page. If you type the NCBI and in the Google and you just click out, and this is the home page that will be opened, okay? So you can go to the taxonomy part here, okay? Taxonomy part, and you can click on to the taxonomy part. Once you click upon, there will be few databases that will be shown. You can just click on the first one, taxonomy page, and that is the page that will have the information related to the taxonomy. Other thing is that, just show you one thing, just give me, just if I go back, there is this literature part also, literature serving. This is also important for many of you. Okay, suppose you want to retrieve any particular information that has been published related to some kind of plant or some kind of fungus or algae. Okay, so you can retrieve information out of that also. So there is this option called literature where you can find publications related to your organisms or like that. So you can use this also for your research. So it's not only molecular biology or something like that. Lot of other information you can use. There is this option called chemicals and bioassays. Okay, because you know that many of the medicinal plants have been used for carrying out bioassay experiments. Okay, so if you are looking for that kind of thing like medicinal properties of plants, maybe you can use this part of the database, that is the bioassay part. This is particularly the PubChem bioassay part. You can go to, and they will, lot of information related to bioassays have been published. So if you have any particular medicinal plant, and if you want to see if that plant have been used in some bioassay experiments or not, you can use this part for looking into that. Even the PubChem part is also important. PubChem is a database part where it will have information related to chemicals, any kind of phytochemicals also. If you want to look for any kind of phytochemicals that are commonly found in plants, so you can use the PubChem to look for that kind of chemicals. If you just write in the Google PubChem, it will be directly shown to you the page, and you can search for any kind of chemicals that are associated with plants, maybe some phytochemicals or maybe some steroids also, maybe some metabolites, secondary metabolites. If you want to look into the chemistry of that, that is also helpful for the PubChem. So it's not only the genetics and molecular biology, but a lot of other important aspects are also there in the NCBI database. Okay. Anything more? Anything more? Anyone has any more question to ask? Okay, if not, then maybe, sir, are you there?
Yes, madam.
Yes.
If anyone wants to ask any question, please go ahead.
Yes,
because
if anyone
it's a
wants
wonderful
to.
opportunity for all of you.
I think I have to take another class in the next week, so I'll try to cover the other aspects. Okay. Yes, I'll try to cover the other aspects, that is the sequence alignment and phylogenetic part, and I will also show you the structure prediction part.
Okay, madam. At the same time,
so
timing will be the same?
Yes.
Okay, fine. Then I'll prepare the link.
Yes, sir. Maybe, sir, you can create a link and send me an invitation. So I think on 18th I have another class.
Okay. On 18th.
Sir, yes, sir.
Okay, sure.
I think I received a letter that I have to take two classes.
Yes,
on 11th and 18th.
Okay.
So maybe 18th at 5 p.m. I have to take another class, maybe the next Saturday. Okay. So you can send me the link. You can ask your ma'am to send me the link, and I can join with that same link. Okay, sir?
Okay, madam. No problem.
Okay, I think if no other question, then I might end my class today. And if you have any queries also, you can always email me anything related to your research, and we'll be happy to help you at this point. But maybe when you are in your PhD thesis or something, so at least you will at some point or any point you might have to use these databases for searching information. So that is important. So how do you retrieve the information and in what format? So you should know these parts. Okay. So, sir, with this, should I end the class?
Yes, thank you, madam. Thank you so much for this informative lecture. And I think we will be hearing you next time. Okay, madam.
Okay, sir.
Thank you.
Okay, sir. Thank you, sir. Okay.